parasite protein. Hedda.
Thank you, Antonio. Hello to everyone around the world. I'd like to thank Antonio and Caetano first for the invitation to present our work here. My lab has a long-standing interest, as Antonio mentioned, in the quality of B-cell responses in humans, starting with autoimmunity, and recently we moved to the malaria parasite Plasmodium falciparum. This parasite is a very complex organism. It's a unicellular parasite with over 5,000 genes and a very complex life cycle, which is depicted here. It's transmitted to humans through the bite of infected Anopheles mosquitoes, which inject a very small number of the so-called sporozoites, this stage of the parasite that's depicted here, into the human skin. Around 10 to at maximum 200 sporozoites are injected. Then within a very short time, minutes to hours, these sporozoites move from the skin to the liver and then infect hepatocytes. This phase of the infection is still asymptomatic. It takes about 1 week until these parasites rupture the infected hepatocytes and then are released into the bloodstream. They are then called merozoites, and they look very different and express also different genes at this stage. And these merozoites then infect repeatedly red blood cells and develop in them, multiply in them, and then burst open. And this phase, is the enormous multiplication of the parasite, is associated with malaria disease symptoms that can be life-threatening if they are not treated. And at the same time, during this phase of the infection, also sexual parasites develop, and if they are taken up by the mosquito, then the life cycle can be closed again. Dozens of malaria vaccines have been designed that target all different stages of the parasite life cycle. The pre-erythrocytic stage here to prevent the infection, but also the erythrocytic phase to prevent disease symptoms, and also the sexual stage to prevent transmission to the mosquito vector. However, so far only a single candidate made it successfully through clinical phase 3 trial, which was conducted in many African countries. The vaccine candidate is called RTSS. It comes in an adjuvant that is called AS01. And this was conducted with 15,000 kids, but it showed relatively low efficacy of less than 50% and also short-lived protection, suggesting that this vaccine should be improved in order to mediate good protection. And so that's a question that we became interested in. Is it possible to improve a CSP-based vaccine? This vaccine. So this vaccine, I haven't told you, the target is targeting circumsporozoid protein, which is the major surface protein on this sporozoid stage of the parasite, so the stage that is transmitted by the mosquito. And this protein has been shown is essential for development of the parasite in the mosquito, but also in the mammalian host. The protein consists of 3 domains. There is an N-terminal domain and a C-terminal domain that flank this very characteristic central repeat domain that is composed of many 4 amino acid motifs, these so-called NA and P motifs. And these motifs are 100% conserved in all Plasmodium falciparum parasites that have been isolated so far. There are around 38 of these in this domain, and it has been shown that antibodies against these NAMP motifs mediate protection, which has been shown in animal models already many years ago. And based on this finding, RTSS, this vaccine candidate, has been developed, which contains 18 of these NAMP motifs and the complete C-terminus of the CSP protein. So this part here, in a fusion protein with the hepatitis B surface antigen, and also complex with additional hepatitis B surface antigen to generate virus-like particles that are hopefully more immunogenic than the recombinant CSP protein itself. So this is the basis of the vaccine. And so what we decided to ask first, before we went into the design of novel vaccine strategies, is to go back and ask what is the overall quality of human anti-CSP antibodies, because at the time no human antibody against this protein had been described. We also wanted to understand what are the molecular characteristics of the most potent antibodies that we could hopefully find, and then also to understand how the antibody response against this protein evolves in humans. To address these questions, we collaborated with clinical partners at the University of Tübingen, who at the time were running vaccine trials against malaria that were based on the injection of live parasites. So this sounds very weird, but it's sort of a standard in the field that humans can be exposed to live parasites here at very high dose, 50,000 sporozoites at a time. These volunteers are exposed 3 times, 4 weeks apart. And this is possible if at the same time they are undergoing chemoprophylactic treatment, which basically targets the parasite when it ruptures from the hepatocytes, and then the parasite gets killed. So these individuals have parasite development up to the stage where the parasite comes out of the liver, but then the parasite is killed by the drug, and there are no disease symptoms. So this setup here allowed us to study the response over time, and we obtained blood samples always at day 7 after each parasite exposure. This is the Malaria vaccine. And then we made monoclonal antibodies from circulating CSP reactive memory B cells that we isolated by flow cytometric cell sorting. We used an Alexa labeled CSP bait. Now you can see there's a small number of CSP reactive memory cells that are seen in the blood of the immunized individuals, and the frequency goes up with repeated exposures. Here's always 1, 2, 3, corresponding to these numbers here. And from these cells, we made over 400 monoclonal recombinant antibodies, and then tested for binding to CSP in an ELISA assay. Very simple here, is seen here. Each line corresponds to 1 antibody. And to get an idea of the binding strength, we plotted the area under curve of antibodies that we obtained from memory B cells that were isolated at the first exposure, after the second or the third parasite exposure. And as you see, the quality of the response increases over time. Most of these antibodies targeted the NANP repeats as expected. And here you can also see that the binding strength improved over time. So this suggested to us that these cells probably underwent affinity maturation. And indeed, we observed clusters of clonally expanded B cells that had accumulated somatic mutations. We could build these nice trees here with the matching, based on the analysis of matching heavy and light chain VJ genes and same CDR3 length. So, however, when we looked at the number of mutations in the antibodies that we had isolated and looked at the CSP binding strength, again, measuring the AUC here, we observed that the vast majority of the very good binders were actually from antibodies that had very few mutations or were even unmutated. And these antibodies accumulated, or B cells expressing these antibodies accumulated with time. And there were many seen after the 3rd exposure, whereas the antibodies that were more mutated, many were seen already very early on, suggesting that these were pre-existing memory cells that got recruited into the response. But very few were actually very potent binders. So this was interesting to us, and we wanted to understand more about the gene features of these germline antibodies. And when we looked at the gene features of several of these antibodies that showed actually really high affinity or good affinity to the NANP epitope here, we found that many were expressing the same or using the same VH-V light chain gene combination. So always a 333 VH gene segment in combination with the V kappa 1,5. And it was also striking that the kappa CDR3 length was mostly limited to 8 amino acids. And this is unusual. And so we wanted to link this to parasite inhibition and tested these antibodies in an in vitro assay that measures parasite traversal through hepatocyte cell line. It's a standard assay in the field, and we observed that not only were these antibodies very good binders to CSP, but they were also highly enriched among the very potent parasite inhibitors in these assays. So in total, we observed, we identified 63 antibodies that we found in 4 out of 8 individuals from which we cloned antibodies. And I mentioned most of these antibodies were unmutated, but some had mutations. And so when we looked at the mutation profile in the heavy chain and the light chain, we observed hotspots of mutations where we saw this enrichment of mutated residues, 3 in the heavy chain, in the CDR1 and CDR2, and 1 in the kappa CDR3 region. So we want to understand how these mutations improved antigen binding, and we performed assays where we either had or did not have the mutations in the antibodies, and we were surprised to see here that actually 2 of these mutations at these 2 positions increased the affinity to the NANP, which is what we expected. However, these 2 mutations here, H56 and CDR2 of the heavy chain and kappa 93 in the CDR3 of the light chain, had actually no effect on antigen binding. So this was weird, and we didn't understand it in the beginning, because the selection should be based on affinity to the NANP, according to textbook. And so we collaborated with Jean-Philippe Julien, a structural biologist at the University of Toronto and the Hospital for Sick Children. And Steve Scully in his lab obtained a co-crystal structure of one of our antibodies with an NANP5 peptide here. And you see in this co-crystal 2 Fab fragments actually bound to this peptide here, one on the top and one on the bottom here. And what Steve observed is indeed these 2 residues, H31, H50, mediated direct contacts with the peptide, whereas the other 2 mutations mediated interactions between the 2 Fab molecules. And so this was the question, would this improve antigen binding? And to test this, Steve used ITC measurements, and he could show when he compared the wild type antibody and a mutant that he generated, which did not allow these residues to interact and to mediate these Fab-Fab contacts, that then binding to an NANP5 mer peptide was inhibited, whereas binding to an NANP3 peptide, which allows binding of only 1 Fab here, then this mutation had no effect on the binding compared to the wild type. So suggesting or demonstrating that these Fab-Fab interactions indirectly improve binding to this repetitive antigen. Now, what is the reason for the selection? We assumed that these mutations should improve BCR signaling in the context of a cell. So Katharina Imkeller, a former PhD student in my lab, generated cell lines that expressed either the wild type antibody on the surface or this YY mutant, which can't engage into these homotypic Fab-Fab interactions due to sterical hindrance here. And then she stimulated the cells with this NANP5 peptide and measured calcium flux. And as you see here, this is the flux in the wild type cell line. It's much stronger and faster than for cells that expressed this YY mutant, where it was significantly impaired. And Katharina could also quantify these measurements. And she found that the frequency of activated cells was reduced, and also the median activation time was much longer for the mutation that could not engage into these homotypic Fab-Fab interactions. Now, I've shown you that potent antibodies against this central repeat domain were frequently encoded by these VH333 antibodies. They underwent this homotypic affinity maturation, also improving antigen binding indirectly. But the question to us was, are there any other B cell epitopes in the CSP that could be protective? And so we looked at the N terminus and the C terminus, and I'm just summarizing a lot of data here. But the bottom line is that we identified only a few antibodies that would recognize the C terminus of CSP, and these had no functional effect, most likely because they couldn't bind the parasite or the CSP on the parasite surface. And antibodies against the N terminus we could not identify. And the only antibody that has been described against the N terminus, which is a mouse antibody, did not show any function in our assays. So this left us with one part of the protein, which is called the N-terminal junction here, that contains some of these NANP motifs that are interspersed with a very similar NVDP motifs and also an NPDP motif here. And Antonio Lanzavecchia has actually shown, together with Bob Cedar, that this region of the protein contains protective B-cell epitopes, and that these antibodies that bind these epitopes are often cross-reactive also with the classical NANP repeating motifs here in the central repeat domain. And so this just shows you how similar these epitopes are, or these motifs are. And there's also very similar, another similar motif in the C terminus, a so-called NANA for amino acid motif. So we wanted to understand whether our antibodies would bind to the junctional epitopes, and we tested this against a panel of overlapping peptides, short peptides that are shown here, and also just the NANP and the complete C terminus in an ELISA. And this is shown here. And as you see, many antibodies, or most of them, bound the NANP motifs, but at similar frequency, NVDP motifs were recognized, corresponding to this peptide, and also the other peptides were recognized, suggesting that many of these antibodies are actually cross-reactive, as had been described. And so when we look at the binding profile of individual antibodies that is shown here, then we see that many antibodies here, or some bind only a single peptide, shown here, but actually the vast majority shows cross-reactivity to one or two or more of these peptides as depicted here. So the vast majority of CSP antibodies are actually cross-reactive with these junctional epitopes. And so the question was, what discriminates antibodies that are specifically recognizing only one peptide, NANP for example, to the ones that show cross-reactivity with some or more of these junctional peptides? And so we measured affinities here. This is work done by Roger Gopal-Murugan. And what he showed is that the cross-reactive antibodies had overall higher affinity to the NANP motifs, but also to the junctional epitopes here, suggesting that it's not just specificity, but cross-reactivity is also linked to affinity. This was then the question, how does this link back to parasite inhibition? And so we compared antibodies with high parasite inhibition and looked at their affinity to the different peptides. And the one thing that discriminated is that high affinity to the NANP, the classical motifs, but not to the junctional epitopes, was linked to parasite inhibition, suggesting that this NANP is still a very important epitope. and that affinity to this rather than to the junctional epitope is relevant for parasite inhibition. We also determined IC50 values for some of the most antibodies, most potent antibodies, and observed the same thing here. And we also tested some of the antibodies that had very different binding profiles, not only antibodies that we described, but also that some that have been reported in the literature in an in vivo model. And here again, we observed that the antibodies with high affinity to CSP, no matter which epitope they preferred, either the NAMP classical motif or the junctional epitopes, showed very similar levels of protection. So this brings me to my summary. We think that so far all known potent CSP antibodies target the central repeat, including the junction, whereas antibodies against the C or N terminus that are potent have not been identified so far. And we think that epitope cross-reactivity is associated with high affinity and thereby protection, independently of whether the antibodies target preferentially the central repeat or the junctional epitopes. So what does that mean for vaccine design? We believe that we need to improve the quality, meaning the affinity of the anti-repeat junction response, and most likely by promoting germinal center reactions and affinity maturation. And with this, I would like to thank the people who were involved in this work. I mentioned Rajagopal Murugan, also Katharina Imkeller, who was helped by a number of people in the lab that are now still in the lab or former students, and also our collaborators, Jean-Philippe Julien, Steve Scully at the University of Toronto, and Elena Levashna and Julia Costa, who perform all the parasite work at the Max Planck Institute for Infection Biology in Berlin, and our clinical collaborators. And with this, I'd like to thank you for your attention. I'm happy to answer any questions in the QA. Thank you.
Thank you, Hedda. This is a very important work because malaria is still